Hi, welcome to week 5. This week I'm going to take you on a little tour through your immune system. And here we begin with the innate immunity. Okay, so by the end of this section, you should have a rough idea of the body's innate physical and chemical defenses, the inflammatory response, and you don't have to mind the complement system too much because that is, well, except to know that it's there and have an idea what it does. There we go. We have the ink. It's kind of a bridge between a couple of systems, but I'll get there when we get there. Okay. So first up, innate immunity can be separated into two main categories. We have the very passive physical barriers right here, and then we have the internal defenses. Now later on, I will go over the adaptive immune system about antibodies and the humoral response, cell-mediated immune response, memory, right? But so those are the innate and adaptive are the two main components, but under innate, which is what I'm talking about now, we have two major components because you can't forget the physical barriers, right? Your skin, hair, the cilia in your throat, well, in your upper respiratory system, right? Why am I saying right? I don't think we've talked about this yet. All right. So your mucus gets brought up from your upper respiratory system. That's one of the things that gets impaired when you smoke. So it makes it you more vulnerable to respiratory infections. And let's see. So mucus in general, digestive enzymes in the mouth. Now we did go over some of those, right? Do you remember any of those at all? And then, of course, the stomach, right? Stomach acid. There was one bacteria I may have mentioned that can survive well in there and cause a problem. But other than that, you're looking at a pH between 1 and 2, right? Which, remember, that was acidic, and it's very acidic. So you can't ignore your physical barriers. Now, you could argue that there is a passive layer on top of your skin, and that's your normal flora. You can tell I haven't practiced with this yet. Much normal flora. And those are the bacteria and some fungi and some mites and other parts. But these guys, here's an example. Whoops. Of a bacteria. And that would be Staphylococcus aureus. No, wait. Well, sorry. There is a Staph aureus that can be non-pathogenic, but epidermidis is what I'm thinking of. Now, it's not that they're intentionally protecting you. It's just that when your skin's already well populated with them and a pathogen tries to come in, they are going to have a harder time doing well because, the N is full. There's no room for the pathogen, the would-be pathogen, right? So anyway, physical barriers and internal defenses. That's the takeaway here. The internal defenses include phagocytic cells, so those are eaters, of foreign cells. There's complement proteins, and we have the inflammatory response that kicks a bunch of this off, and we have natural killer cells there. So the inflammatory response. So white blood cells, able to move. So monocyte will circulate in the blood and the lymph. They'll develop into a macrophage or a mast cell. And in connective tissues, they can release histamines. So that's for an allergic response, will induce an allergic response. And then cytokines released when a pathogen is recognized. And that's what we're looking at here, cytokines. There we go. Whoops. Yeah, it still have ink on. Okay, good. And so when a pathogen is recognized, the cells will change into different versions, and you didn't really talk about all that, or what the earliest cytokine is. Basically, cytokines encourage inflammation, and that's what helps this immune response to really get mounted. So you'll have white blood cells that will get signaled to stop and come in. If you remember that inner life of the cell video, that's what one of these cells were doing. Extravasion. They were moving from circulating blood, into the tissue by passing through epithelial cells making up the vessel. So neutrophils, they arrive early, and they're the most abundant. And we have natural killer cells. There we go. So these are cells, lymphocytes such as NK cells. They have large nuclei. They actively absorb the right stains. This is how you can identify them, in a histological assay of a blood sample, right? So these guys identify infected cells, like with a virus, because, so cells, normal healthy cells will present a sample of themselves as like their passport or their papers or their ID, if you will, to interrogating immune cells like natural killer cells. If everything looks normal, then the natural killer cell will move on. If the cell presents something abnormal, and this is a major histocompatibility, this is MHC class 1. Oops. Well, okay. From cells at work, that's the natural killer cell character. But so we have major histocompatibility, MHC class 1 recognition. So a healthy cell will pass this check, and the unhealthy cell will not. It could be cancerous, it could be infected. Obviously, some viruses are able to get around this, and you'll have cells that either will not display any antigen at all, any MHC at all, or they will have a fake one, right? There's a lot of interesting molecular defenses out there. So if, however, the cell's recognized as being aberrant in some way, then the natural killer cell will kill them, by inducing cell death. So that's the NK cell, part of innate immunity, right? Here are some other actors. Well, here, we'll go ahead and skip to this part. Again, I wish I had a way to show you cells at work. The English translation, the English dubbed version. There is, maybe I'll just make those available anyway through Edpuzzle. But the Japanese one with English subtitles are available. But this is the cast from cells at work matched to their cell type. So the first one you meet in the series is the white blood cell. They call him that, but he's a neutrophil. And there's macrophage, monocyte, NK cell. So like I showed you before, natural killer cell, and then the mast cell. And I'm not going to go into what they all do. I do encourage you to watch it. Again, I couldn't find a way to download it. But okay, so that'll about do it for that section of innate immunity. I just had a difficulty. I was talking to myself. The recording just stopped on me. I mean, I'm talking to myself anyway, but hypothetically, some of y'all are going to watch this here later on, right? Okay, so innate immunity. So now we're going to move on, and I'm going to describe adaptive immunity. And now I can pull up this other thing with the notes. Oops. Okay. Adaptive immunity. See, there's writing and stuff over there already. Anyway, by the end of the section, you should be able to have something to say about adaptive immunity and maybe describe the cell-mediated immune response and humoral immune response and describe immune tolerance. Now, adaptive immunity centers around recognition of a pathogen or something, an abnormal cell. And that circles around the use of the antigen. Convert to pen. There we go, pen. Antigen. Yay. So the antigen is like if we draw the virus as a sphere thing and it's got spikes all over it, these spikes tend to be the thing that is able to be, it uses it to adhere to the cell, its target cell, but it's also what the body can use with these antibodies. Oops. Well, they generically look like this, right? Or they can bind stuff here. And once you tag something like this, you can activate a bunch of other responses that will hopefully take care of the invader, right? That's the idea. So our two components of the adaptive immunity are the cell-mediated response, immune response, and that's primarily by T cells. And humoral immunity involves B cells and antibodies, right? Okay. Yes. Well, let's go on here. B and T cells. So B cells, they mature in the bursa, and T cells mature in the thymus. Now, while they're maturing, they're supposed to be pruned out if they are recognizing your host cells, your cells, your own cells, self. And if the immune system works properly, that's what it'll do. In autoimmune disorders, this has gone wrong in some way, and you have immune cells attacking attacking your own tissues. So here is a T lymphocyte by electron micrograph, and the T and B cells are indistinguishable by light microscopy. And but if you watch cells at work, then they look like this. So we only saw, unless I missed something later on, a couple of memory cells. We had the memory cell, memory B cell, and then the B cell itself. This is the one that was shooting antibody all over the place. And now for T cells, we have a collection of them here: the killer, memory, naive, effector, helper, regulatory T cell. And again, not all of you can watch this or are inclined to, so I'm not going to belabor it. There we go. So the antibody that I mentioned, and I drew a poor cartoon representation, it actually has two binding sites. So again, remember, articulated magnetic Legos. That's in essence what the majority of molecular biology comes down to. But you'll have a binding site. Now these are generated either by memory, initially they're generated randomly by B cells. Random B cells make random antibodies. If they don't bind to something and cause downstream reactions, then they're discarded eventually. But yeah, so the antigen again is something generally on the surface of the pathogen, in the case of pathogens, that could bind randomly with a matching antigen binding site. So B cell receptors are a type of antibody, and they're embedded in the membrane, and they have these are variable regions. This is the fixed region. That's the heavy chain. That's the light chain. Anyway, you don't need to know that part. Okay, so anyway, when an antigen binds on the B cell to the embedded antibody here, the antigen molecule is brought in, so it's basically eaten and digested a little bit, and then it gets presented. So before I mentioned for the natural killer cells, for innate immunity, that was MHC type 1. This is, can you guess? MHC type 2. So the B cell presents this antigen, and so now it's sensitized. So let me see, sensitized. But then it needs to encounter a helper T cell. See, that was this dude down here. Oops. Okay. I don't know why. Okay, sorry. No. I'm just messing it up now. Anyway, a helper T cell to become activated. And when it's activated, it will, the B cell will be able to start producing, or they'll become a memory cell. They either start making tons of that antibody that it's, the antibody that matches that antigen, or it'll become a memory cell and become inactive. So, and then either secrete like crazy, and that is a plasma cell, or it will be a memory cell. And I know you're all like, well, exactly what's the mechanism that determines the fate of any particular cell? I don't know off the top of my head, and that's a little further than we need to go right now. We're skimming over the surface of this very deep subject. Okay, so, right, the plasma cells can produce like, oh my gosh, was that right? Yeah, the plasma cells produce and secrete large quantities up to 100 million molecules per hour. All right, so here are the basic classes of antibody. Usually the one that we're talking about with pathogens is called IgG. There's the secretory IgA, and that's usually in, is that not on here? In breast milk. Yeah. And we have IgD, IgE. So did I say, yeah, IgD is the B cell receptor, and then IgE is involved in allergens and against parasites. So these pentamers, so you got like 5 of these all stuck together. They, that's more detail. The majority of your circulating antibody is going to be the IgG right here. And that's after a pathogen has been encountered before. You can have the antibody raised against that particular pathogen. Okay, moving on. Here we go. So, how do these antibodies do? What do they do to their targets? Well, first they can neutralize them, and that's just by blocking their biz. So this virus, if it was in 3-dimension and had antibodies coming off of here too, right, of all sides, it's not going to do a very good job binding with its target cell because it's being blocked up. And see, there's diphtheria toxin. If the diphtheria toxin can't interact with its target because it's got covered in antibodies, then it's been neutralized. So another version of this is when it's all tagged, you can get opsonized through opsonization, and that's where it gets consumed by a macrophage or a neutrophil. So it's basically tagged for destruction. And then I mentioned it before, the complement activation, and that's where a pore gets made inside the cell and it leaks out. It's really pretty cool. I think so. I think it's pretty cool. Okay, so cell-mediated immunity. So here we go. So unlike B cells, T lymphocytes are unable to recognize pathogens without assistance. Instead, dendritic cells and macrophages first engulf and digest the pathogen, and then they have these antigen-presenting cells will detect, engulf, and inform the adaptive immune response about an infection. So the dendritic cell, and again, I didn't pepper those characters all through here because not everybody's up to speed on, or is willing to get up to speed. My 10-year-old kind of hates it. Cells at work. I thought he would like it. He enjoys Naruto and My Hero Academia. Is that the one? Not, yeah. Anyway, he does not. All right, so anyway, the dendritic cell is the immune cell that will mop up antigenic materials in the surrounding and present them on its surface. And so dendritic cells inform, and that's the guy in green. But again, I got to stop circling back to that show. Dendritic cells are located in the skin, the lining of the nose, stomach, intestines, and which makes sense, right? Because you're going to encounter pathogens at the barrier. Okay, once they become activated, the dendritic cells, they become APCs. They migrate to the spleen or lymph node. Macrophages can also do that. So again, here's APC, right? Engulfs the foreign antigen. Here's our cartoon. And I think we're saying this is tagged. Yeah. So, and then it gets digested, and then it gets presented. Right, so now they are antigen-presenting cells. Okay, so here we go. What I was just mentioning here. The helper T cell interacts with the APC, and becomes activated. Now, after it's activated, it can release cytokines. The cytokines can activate B cells and other T cells, including, well, here, this is a T cell clone. It now is a killer T cell, and it's involved in the cell-mediated immune response. Here's how they work together. So the T cell, the helper T cell, so I mentioned the B cell before, can induce the B cells to clone itself, and either become the plasma cell or the memory cell. And type 1, type 2 help, no, okay. Memory, immunological memory. Hopefully you all have some of this action here. There's the antigen on the bacteria. Okay, so again, the B cell, the antigen will bind to its receptor, which is an antibody, remember IgD. The B cell internalizes it and presents it on its MHC class 2. And the helper T cell will recognize that and then activates the B cell. So we have the B cell, random receptor sites here, random variable regions. This one happens to match something on the outside of the bacteria. And it's mediated through the, you don't need to worry about knowing the CD complexes here. So the type 2 helper T cell releases cytokines, and now we have memory B cells and plasma cells. So this is the primary immune response. This is when it's just first encountered. So primary immune response. Wait a second. What was that? Wait, this does do levels? Okay. Anyway, primary immune response. So once you have had a primary interaction, then if you encounter the same thing later, you'll have a secondary response. So let's compare what those two look like here. So here's our primary response. This is kind of generic. So remember that IgM there, 20 days, we get the IgG levels really high. Initially, it's IgM. And that was the pentamer. Sorry, I just, got to, yes. Double check myself. All right, so this is your first exposure. It takes a long time for your antibody response to start being mounted, and then it comes in pretty strong, right? So say you've recovered and you encounter it again. So now this secondary level, because of those memory cells, remember our BMEM, they're able to start differentiating into plasma cells rapidly because you already have a population of them. And they start secreting, pumping out tons more. See, roughly, I don't know. Well, okay, this isn't supposed to be the scale. So I shouldn't be trying to proportionalize it. Anyway, so the secondary response occurs a lot faster, and higher levels of antibodies are more sustained. And that's mostly IgG. Now, the B and T memory cells will eventually die off if the pathogen is never encountered again. So that's where we talk about immunity can wane. Well, here's the news. Even if you've had the actual disease and it wasn't a vaccine, that can wane as well. Okay, so the lymphatic system, briefly, this is our kind of our immune system information highway. So lymph is the watery fluid that bathes tissues and organs. It contains protective white blood cells, but does not contain red blood cells. Lymph moves about the body through the lymphatic system, which is made up of vessels, lymph ducts, lymph glands, and organs, like your tonsils, adenoids, thymus, spleen, these things like that. Cells of the immune system can travel between distinct lymphatic and blood circulatory systems, which are separated. And that's extravasation again, like that video, the inner life of the cell. And that's enough. So a lot of these antigens can accumulate in the lymph nodes. And that's one of the things they check in cancer to see how far it is spread, is if the cancer cell is detectable in lymph nodes. Because a really successful or bad, cancer has, migrated. All right. Meanwhile, the spleen houses the B and T cells. They live in here, along with the macrophage, dendritic cells, and natural killer cells. And this is where the antigen-presenting cells will go to communicate what they've learned, like the dendritic cells was an example of that. So the spleen is to the blood as lymph nodes are to the lymph. This is where training happens, right? Training for fighting potential pathogens. Okay, I think this is long enough for this week. I guess I can split this up into next week, looking at disruptions in the immune system, and then a lot more closely. So this is where can talk about some things like really bad allergic reactions or autoimmune disorders. And let's see there, hypersensitivity and autoimmunity. And then after that, I think if it might be our last week after that, maybe we'll do a little space stuff, since that's in the name of the course. And maybe we'll take a virtual tour of our solar system or something. All right, so have a good week. I'll have some assignment stuff up. Hopefully you can knock that out really quick. And I will see you. I'll be in touch with you. You can contact me on my Google Meet every day. All right. Have a good week. Bye.